Hi, Louisa. Can you unmute? Thank you.
Sorry about that, Jermont. Didn't realize it would remute. Okay, so hi everyone. I'm Louisa Russell. I'm your in-person moderator for this session, and we have Jermont Chen online as our virtual moderator. So hopefully people online, if you want to participate in the discussion, throw it in the chat. Jermont will pop in with some comments from you guys. So I guess in terms of this session, the first breakout was really to kind of look at blue skies analysis, what could exist if we just dreamed about whatever we wanted to exist. I think this session is focused a lot more on concrete connections and opportunities. If you were to think about something that either you're working on and you have a problem with already that you've run into, or that you know of that other people have run into, where are places where these two fields can make some connections to solve problems on both sides? Whoever wants to start with their most tough problem.
Yeah, so in my research now, currently, a major challenge is to find a reliable aging model. Really, the in vitro, I think you all mentioned
Yep.
that as well. Yeah, like what kind of aging, either cellular model or tissue model that we can study in vitro, that we can manipulate, we can watch, we can track, we can model. So yeah, it's hard to find this kind. There is, of course, the cell synthesis, which we can track, but it is there is debate about whether it is really aging process.
So when you say that you need a reliable aging model, are you thinking in terms of like a 3D model?
Or even cellular model,
Okay.
just single layer cell model. That's as long as that people in the field will accept, oh, this is a good model for aging. Because this is hard, right? I mean, it is. So because now I'm moving my research from yeast to mammalian cell, and a major question we have is that what kind of model system we should start with, and related to your research, actually we are now starting to study blood stem cell, but it is more of an in vivo model, really. Like, I mean, we can track the cell out for a little bit, watch it for a week to the most. And yeah, according to our collaborator, after a week, people don't think this is stem cell anymore. So
So I have so many questions. So you're tracking a stem cell in the bone marrow,
yeah,
and like an HSC? An
HSC.
HSC in the bone marrow, and
Yeah.
you're tracking that over time as the mouse grows up into like a.
No, we just, I mean, currently what we try to do, because we have microfluidics,
Okay.
time-lapse microscopy, what we really do is,
Moment in time.
that's right, treat it as yeast, like we extract them, purify them, and then culture them on a single layer, look at the division for a week, and we can put in some markers that we think are important for aging, and then study them. I mean, basically, if we can look at dynamics for young mice
I
versus
see.
aged mice, and if we see some different dynamics, we can perturb them, we can see whether we can reverse the process. Yeah,
And by dynamics, sorry, you mean differentiation? Like the changes, or what do you?
differentiation is one of them, for example.
Oh, right, we
I know, I just realized we needed to take
Oh.
a quick pause. You don't have to be. Sorry, it was just like maybe someone has organically decided. No, that's totally fine. Okay, so we do need someone to volunteer to be the note taker / presenter, if somebody wants to be. Doesn't have to be you. I didn't assign you. Any volunteers? Do I need to assign someone?
So. Do you think it should be a trainee, or do you think it should be a PI?
Doesn't have to be.
I'm slowly trying to weasel out of it, but. But I will do it. For the love of the team, I will.
Well, it's less about strict note taking and more about being excited about presenting this part to the rest of the group.
It's also very no stuff.
Awesome. Thank you so much. Sorry about that. Continue your discussion.
Yeah, by tracking, what I mean is, for example, these aging hallmarks, we can look at the metabolic activities. We can look at
Yeah.
the gene regulation, using fluorescent markers. Yeah, but it is not as good. I mean, it's still mice model, right? I mean, just get cell out
Right.
and watch a snapshot, a
Yeah.
very short period of it.
Well, what's hard with the HSCs, and I know this because I work with them, is that once you remove them from their environment or their
That's
in
right.
the matrix, they putter out pretty quickly. So, but maybe that's the perfect model because they senesce, right?
Yeah.
So.
That
Yeah.
actually sounds like a good model.
It's not a good model, because well, the problem is they either spontaneously differentiate or they can senesce. And I mean, we haven't been able to control which way it goes, but if you want to keep an HSC, which is what we want to do, it's really hard.
Yeah, and the in vitro senescence, is that a reflection of what happened in vivo, right? I mean, that's what
Yeah.
people really care about.
Yeah.
Because you can always see something change.
Yeah.
But that's meaningful.
Yeah, I mean, so there's two things just building off this, and one, like the SynBio is oftentimes, very often, a hammer running around looking for a nail. I mean, that's fine. I'm a hammer looking for a nail in a lot of situations with research, but one of the appealing things about adoptive cell therapy is that the cell product is individual cells. Okay, so you can measure, whatever you're engineering, you can, same with yeast, right? On an individual cell basis, right? It's not, and so you can very easily parallelize what you're looking at across a population of cells or across wells. I think that I would love to get into aging research if we had models like this, where I could say reliably test a genetic construct in a relevant model. And I think if I'm personally, maybe selfishly, looking to come away from this meeting to maybe find somebody that has something like you're looking for, like maybe an organoid model, or maybe some very simple like tissue engineering type model where they put cells on a construct, differentiated them to some extent, and then we can look at many of these, because you really can't turn the crank on synthetic biology unless you have the ability to iterate in parallel. And then if you have the ability to really parallelize stuff, then other stuff opens up, like machine learning, right?
Exactly.
So, yeah.
I think because we are from the same perspective, actually.
But what do you do in yeast? Can you do it in higher organism or different model system or even senescent cells?
We can do it in cell senescence, I think, like if we believe, like take the primary fibroblast, culture then in vitro is a good model. I believe we could do that. That's not
But
difficult to do.
I just learned in the last session that you can induce, senescence. You could almost take your HSCs and have a global senescence sort of event with your entire cultured cells, and then maybe have one that's not, just by irradiation or whatever there's chemical means as well. And so, I mean, that might be. It's a very basic model.
Yeah.
Go ahead, Mark.
Yeah, and because the next question is because the reason that we want to understand what's going on there is that we can then hopefully design synthetic circuit or the other approach to reverse it or to slow down the process. But how to?
Yeah, so I have a totally out of the box comment to make. Have you, there's a really interesting paper that was recently published about in vivo reprogramming of hematopoietic stem cells. And that actually makes me think, and we've also shown that we haven't done it in vivo yet, but we can also use our platelets to do reprogramming of target cells. But I wonder if there's if there isn't like some sort of in vivo reprogramming that you can do in the bone marrow, because there's a couple of papers that are I'd be happy to send to you that have recently done this, and it's really interesting. So I wonder if there's a way that you can do something like that with the circuit or something, and then push certain cells to go one way or the other, have maybe the same age mice, that way you have a controlled system in that sense, but then like push in one, like if you have a hypothesis like, oh, I think this gene is responsible for aging faster, or then you can maybe do something like that, because it is hard to remove HSCs from the bone marrow and expect them to do what you want them to do in general.
That's right, that's right.
For like more than 10 days.
Yeah.
So that might be an interesting.
Definitely. So the reason that we want to work on HSC is primarily is because what you said, like we can extract them, we can engineer them, we can transplant it back and see the animal change. So that's the appealing part of the HSC.
Yeah, no, I love HSC. That's totally why they're great. They're just cantankerous, they're grumpy cells.
Yeah.
They're hard to work with, but
I think
very powerful.
you're pulling on a really interesting point here too, of being able to use SynBio to be able to build this complex in vitro model of aging that we're looking for. I guess, how many opportunities are there within that kind of space?
Well, yeah, so in terms of an in vitro model, I think it'd be really interesting to be able to build some sort of in vitro bone marrow microenvironment, right? But that requires a lot of ECM. Very likely, you're going to need multiple cell types because the bone marrow is filled with, different tissues, different cells. So I think a complex microenvironment would be cool, and then I think you'd be able to grow the HSCs better.
Okay. Cool.
And I think there'd be biomaterials and cells, is my guess.
That makes sense.
Nice.
Very cool.
That's right. Yeah, I think that's pretty much my and I also asked that the same question in the other discussion section. I hope to get your suggestions is how to implement synthetic biology to slow animal aging. So basically, when we published these several yeast papers, the major question that I always got is how can you help human with that? So I cannot say I put the Sacchard back into a human and it will work. I mean, don't think gene therapy works, right? I mean, it must be some cell-based therapy. So yeah, so let's say we can engineer, we know a Sacchard that can have a longevity effect, at least in some cell types. What can we do with it? We can of course take the cell out, we engineer it, we look at some aging markers, we can see cell become younger, let's say, the best case scenario. Then how to implement them? That's a good
question. I guess I think about it from the standpoint, there's two ways to, there's two ways to stage synthetic biology interventions in living. It's either delivering the DNA program or the RNA program to cells in targeted fashion. And that assumes that the program stays in there over the duration of whatever you're trying to treat, or it stays in permanent, which is a little bit scary. Or you have the ability to replenish it continuously. Right now you can't do that with AAV. You can't retreat people with AAV. There's a lot of attention going into trying to make it so you can. So that's challenging. So the other way is basically to create a synthetic tissue with its own control functions that operates in parallel to an organism's physiology, right? And I think that one's a little bit more, reachable. So I don't know if that's helpful for as far as what you're thinking, but I think maybe you're thinking a little bit in terms of the in vitro model. You're putting the circuit into the cells, it's having some effect on the cell phenotype. And now we want to recapitulate that inside the animal, right? And an alternative is, to try to actually use a cell device, or cell-based device, and it doesn't have to be from the patient, right? It could be something that, it is, allogeneic, right? Then you can implant that into an animal, and then you can, somehow mediate physiology that way. That's more of like a physiological circuit rather than like a cell regulatory circuit. But, as far as doing gene therapy, this came up in the last section, right? We're sitting here engineering circuits, and we're imagining that a lot of the tools that are going to show up to be able to implement these will arrive. There's people working on this very hard. I tend to just go like this and say, okay, the tools for delivery of gene circuits will show up at some point, right? And I'm gonna really focus on kind of the nitty gritty of how to build regulatory circuits. So, yeah, I don't know. I don't think it's not easy right now.
So I wanna, ask you some questions. I'm the note taker, so I have to my job very seriously right now. So you mentioned create a synthetic tissue to mediate physiology.
Yeah,
You know, what do you know? Let's talk about that for a sec, because
what's around tissue?
well, but I think that kind of bringing it to the aging, are you saying almost like a depot that you would? It doesn't even have to be the word tissue, right? Is it
Yeah.
some sort of? Are you talking about like a synthetic? So synthetic piece of fat that you can just like shove somewhere, and then
Yeah.
like
That could
it's gonna like be a motor to do something, like,
That
or
could
you.
be. I'm thinking about like something that carries with it like a control function,
Okay,
right? And most tissues do many control functions simultaneously,
Mhm.
but if you're trying to treat a disease, it might be a smaller number of control functions that you're
like does it
interested in.
secrete stuff? Does it bring stuff in?
It'll
Like-
it'll sense something that is a biomarker associated with the physiological disease state, and then it'll secrete something to mediate the response. And I think when I think about it, I think about like a biomaterial encapsulated device is the kind of the nearest term thing that we can probably get to. You certainly think about implanted differentiated cells too that come from patients' fibroblasts or something. I don't know, but I think that's a little further on.
So this is almost like Star Wars, you have your big ship and then you send the little ships,
Yeah. Yeah.
to like go find a senescent cell or something.
Well, that could, I was thinking more like in Jennifer's talk this morning about immune misregulation, and that changes over time. So, if you want to detect the misregulation early on with a certain type of controller, and then presumably secrete some kind of cytokine to get back to homeostasis, right? And that's going to change over time, right? So you probably want to put a different circuit in at some point. So if you have this implantable device, then you can change the function of the control program kind of, at will, as long as it's like something that's surgically accessible. Right? So that's kind of what I was thinking. And this type of thing is, I'm sorry, this type of thing is happening in the gut microbiome now, right? There's tons of companies being spun up now where there's like a combination of cells and like biomaterial device that protects the cells from the gut microbiome, and they go through and sense and respond to say like colon inflammation or something. So
I'm just writing it all down. So basically, you're basically saying your input's gonna be this immune dysfunction. That's the input, whatever that immune dysfunction is, that would be programmed. But then the processing, the circuit would then say, okay, I'm turned on now, and now I'm gonna secrete whatever, and that's gonna be the pattern review.
perhaps.
Okay,
So what type of cells we can engineer and then implant to patients? Like I know immune cell definitely, but
Yeah.
blood cell, but can we do it for fibroblast or the other cell types? So
Yeah, you can't. There's the one that's has been in clinical trials recently is a cell type called retinal pigmented epithelial cells. So these are things, so these are things that come. They're really good at secreting. A little hard to engineer, but there's actually like a regulatory path to getting these approved, like encapsulated versions of approved. So that's one possibility. I'm sure there's others sometimes. That gets a little.
and I think too, just the
Terramite.
blood cells,
The blood cell,
yeah,
right?
because there's NK cells, they're easily transfusible. And so, CAR T cells certainly are, the talk of the town, the last, 8 years or so. And so there are ways, I think the blood is a really good way.
Right.
Because it goes into circulation, and then you can program things, decorate the cell with targeting, antibodies and things like that to go to a certain location. And that's done with the CAR T cell therapy. NK cells are also, I think, in clinical trials now, and those are a little bit more tolerable in terms of the immune system. So
blood cell is the key.
I
The way
think
to
so.
go.
Unless you want to fix the eye, right? It's your application, so And, you know, the eye is interesting because you could probably have inducers in your eye drops. So to be able to turn things on and off, but, yeah, but I think systemically, I think blood is probably a good way to go.
That's right. I think that's also one of the conclusion from our last discussion. Like if we
Well,
think about systematic aging.
Yeah, and so what I also kept thinking about when you guys were talking is also, can we build a reporter?
Mhm.
Not a recorder necessarily, but a reporter, like, can we, you know, put something into, let's say the bone marrow, because we're interested in HSCs. And they do, and there are studies showing that if you know, exchange bone marrow, you know, then you can get, you know, young, old, young
Mhm.
changes, right? So I wonder if there isn't a way that we could actually send something in there to tell us what's going on, like to report on senescent cells, things like that.
Mhm.
Not like a diagnostic.
Yeah, exactly, exactly. I think that might be interesting too, because kind of back to your comment where you sort of, we need these sort of ways to track
That's right.
cells through the life of an organism. We could, you know, potentially do that with a mouse.
Yeah, yeah,
Um,
it's a good idea.
so write that down.
In any closed loop regulatory system is gonna start off as something that's just an open loop that you can use as a reporter, right?
Uh-huh.
Yeah, so I mean, that, you know, that would be a great place to start.
That's right, that's right. Yeah, yeah, that's a good idea.
So I guess you were talking about this from the perspective of speed of aging. What kinds of things go into that? What would be the kind of potential inputs or outputs that you might be looking for from this kind of implant device, whatever it may be that we're kind of designing in a sense?
What's your question again? Like, I mean, aging hallmarks, the change of aging hallmarks?
Sure, yeah. So you're talking about a really multifactorial process, right?
Uh-huh.
So I guess what are the kinds of things that you would be looking for within this system
Yeah.
that you would then want to react to?
That's right, that's right.
What do you think about heterogeneity? This came up in one of the talks.
Yeah.
So I wonder if there's a way to go in and just see, like, young mouse, old mouse. If there's a way to report on the heterogeneity of the HSCs.
That's right,
specifically.
yeah, that's actually something we are interested in doing as
Mhm.
well. To look at like several aging markers
Yeah,
and the heterogeneity between individual cells and yeah, and whether it will have consequence basically. So it's more of systems biology rather than synthetic biology, but yeah, but that's definitely an important aspect of it.
About a year ago, Davis and Myers, group published papers. They inject, the Yamanaka factor without the c-MYC, right,
Uh-huh.
into the eye.
Yeah.
Eye of mouse, not eye of human. Maybe worse, the aging phenotype. So
That's right.
yeah, so the output is the aging phenotype, the input four factors.
That's right. That's right. Yeah, that's a very hot topic of study.
So those factors reverse the epigenetic aging markers.
Yeah. Yeah. So, and the other thing I'm thinking about how to implement, actually, I'm more from synthetic biology perspective. I'm thinking about how to implement the approach to help slow down aging is what we are also thinking about is trying to engineer the microbiota, or at least some single cell microorganism that we can eat and to help the gut so that help the whole slow down the aging process together. I think that's something we are also thinking about. So that there I know exactly how to implement, right? so yeah.
So I guess that's interesting too, in terms of the cell types that you're looking for in order to be able to shift, right? You were talking about microbiota. We've also talked about immune cells, and we've talked about kind of senescent cells, all of the rest of the cells. Is it an issue of any one of those things could be changed to be able to make a difference for that entire organism, or do you need to be able to hit all three, the whole system? Is there any sense of that?
Yeah, that I don't know.
Starting with one movie card.
Yeah, right. Already pretty complicated.
Career. Yeah, I mean, it's a really good question. It's a really, really good question. Yeah, I think, I think that all of these places are a really good place to start. The microbiota is interesting, but you do have to, it does matter what kind of bacteria you
Mhm.
want to engineer because, it depends on what's there, because usually the host bacteria do better, the sustainability of it being to stay there, so.
Yeah, that's definitely.
If you guys want to kill that topic, I just wanted to ask something slightly.
Go for it. Yeah.
Yeah, I think I heard a lot of a lot of people obviously working on different things on the aging side, and there was a commonality in the slides was there's complexity and there's a lot of different things interconnecting. We just barely touched on this in the other discussion, but it was, this idea of what's the chicken and what's the egg with aging, and where does causality come from? And I shared last time that it was my primitive understanding of it is it's based on instability of the genome, and that everything is kind of downstream of that we've seen physiology. And that might not be right. I don't really know anything about this, but I focused on senescent cells as something that's maybe upstream of a lot of things. And I don't know, again, if that's the right way to talk about it either. I think you remember this discussion, you were there. But I would, again, walking around with hammers, and I would love to know, like, what is, if the aging folks are going to pick one problem for synthetic biologists to work on that they're most interested in, they think is the most upstream, just in terms of, like, we're talking about designing a circuit that can reverse a phenotype, or say designing a cell therapy that can go and actually kill or extract that cell type from an animal. But like what is that? What can we point to? I mean, even if it's just like a collection of things, like what should I go back and read about, or what would you like to work on with me?
So to me, I think naive thinking, to me at least, I think the causal thing is the damage accumulation. It all starts with damage accumulation that just intrinsically will accumulate over time. There are different types of damage, of course, but for example, at least for yeast, it is starting with damage accumulation and with the asymmetric division of mother cell and the daughter cell, so that the mother keep all the damage to itself, so that it can it has an aging process. The daughter cell, the lifespan is fully reset because the damage is all in the mother cell. So it is more, the origin of it, I would think, is damage accumulation, different type of damage, DNA damage, protein damage, and this is just the consequence of life. Yeah.
Repairing damage is hard as an engineering problem, right? It's very hard.
Yeah.
And removing the damage is much easier.
That's right. Yeah, actually, in our last discussion, we also talk about, like, there are two directions to think about this question. One is to be protective, to protect the cell from the damage, or to make the cell more protective. And the other way is to reverse it, meaning damage removal, right?
Yeah.
Even you have a lot of damage, there is some mechanism, maybe you can remove all the damage so that you reverse your aging process.
Detecting the damage and accelerating from.
That's right. That's right.
That's interesting.
Yeah, I think for the aging,
It is. These are all controls.
it is. For the aging, it's kind of, chicken egg, as you said, but I think there is three big targets. One is, chromatin, including, epigenetic modifications and the genetic things, right? So because of overaging, there is the damage, and also environmental factor affected chromatin. So that's one of the major, target area. I think the second, major target are the immune system. Since many aging-related diseases, the defect, kind of when you can trace back to the immune system. So immunity. Then the third one, I mean, in the other session, we were talking about the mitochondria, right?
Yeah,
I
yeah.
think mitochondria is also, a major kind of thing for aging and for many other kind of diseases.
That's really useful. So
So-
would you say most drugs at this point are targeting one of those three that are on the
So-
table, or is that?
First, we don't have many drugs. Second, I mean, for the drugs, we don't know where the target, so.
Okay,
So there is a target, for example, metformin. Most people think, the major target is on the mitochondria, right? Then rapamycin, major target is mTOR.
Sure.
It's a pathway affecting the immune system and the protein synthesis, So I think an easy target for synthetic biology is mitochondria because it's small, simple, maybe, we can design something. And not just for aging, I think, many other fields, would benefit, right?
So,
Then you can break down from mitochondria to the electron transport chain, then further dissect into, complex 1, 2, 3,
yeah,
So-
no, that's a good,
Then you can study ATP production or NAD production.
yeah,
That has something to, that's a major thing for aging, diabetes and thing, That might be an easy area to target.
it's very.
do you think that it would be worth considering making a synthetic mitochondria?
That's quite, that's where, yeah.
It's just the basic function, not the whole
Factor.
complex
So,
system,
I
but
want- yeah.
it's the basic
Yeah,
function.
as you mentioned, it's quite interesting. I went to a, Golden Conference meeting, someone talking about, doing some kind of, you can produce,
Mhm.
artificially, the kind of, the potential, pump potential,
Right.
then affect ATP production. So then, the cell, the mitochondria, can produce its own ATP without,
Mhm.
outside, nutrition, it kind of, absorb like sunlight, then become, make to become, ATP, right? Back to your question, I think, doing synthetic mitochondria might be hard, right? But modulating mitochondrial function should be, quite feasible based on that study.
Mhm.
So,
And then I guess to kind of break that down a little bit, what are the important functions of the mitochondria for aging? And then can we just, because the mitochondria has, I think that it has probably more functions than I know about. But I'm wondering if we could identify the most important functions that the mitochondria do with respect to aging.
I think three things are connected to aging. One is, energy production, ATP, right? So,
Yeah.
but this is true for all the other functions. More relevant to aging is, like the oxidative stress during metabolism, cause, oxidative stress that affect aging, right? Another thing is, NAD production in the past several years, they think, NAD decrease over, age. Then if, you can do some kind of engineering, by synthetic biology, try to maintain kind of NAD production, maybe that would be helpful to aging, right? So, I mean, this NAD, the oxidative stress and ATP are three things related to aging. I think, maybe the NAD is a kind of, easier task.
So in natural aging, what's the cause of this mitochondrial decline? Like, what's the?
There is there are all the kind of, study and hypotheses, for example, the mitochondrial genome kind of mutated, that's, one of the cause, the other are kind of, the functional, so, some nucleotide chromatin genetic changes also affect, the mitochondrial protein because most mitochondrial proteins are synthesized by the nuclear genes, right? So those kind of, the other things, mitochondrial number, mitochondrial morphology, all, start to deteriorate during aging, so those kind of things should be affected.
It's,
How,
yeah, sorry.
I was gonna ask, how much do all these like markers of aging vary throughout different cell types and tissues? Because I think about like cancer, which is something I'm more familiar with, and like every cancer is basically its own disease.
Right.
Like you can't use the same treatment for everything. How much does that parallelize with aging?
Right, so I think, yeah, that's very interesting. I think there are two sides of the coin, right? So one side, different tissues age at a different rate, right? So for example, Steve Harvard's, study using the DNA methylation kind of, marker show, for example, breast tissue age much faster than other tissues, so it showed. But there are also some common characteristics, across tissues and even across different animals. So there is some, things are common, but there are also heterogeneity among different tissues, But they do, deteriorate over time, just, at a different kind of speed, or may hit a different kind of pathway. So.
Interesting.
Yeah, I think there is a recent work in fly aging where they sequence all the organs, the aging process, and see their speed is different, the heterogeneity of it.
Yeah.
Yeah, it's the kind of resource to answer your question, right?
So I guess Tara, it's kind of what you were suggesting, that there's a possibility to create a synthetic mitochondria, just load people up with it, and then they would be, like, is there a targeting issue there that Amparo was kind of pulling on a little bit? Like, would it need to be somewhere specific, or is it something that you could just?
Almost like this
Yeah.
depot thing.
Yeah, right. There's a lot of intersecting things here. You just rub it really hard and just mitochondria goes everywhere, but.
Yeah, that's a good question. I mean, obviously it has to be connected to, like, because like the mitochondria is inside of the cell, right?
Right.
So the cell actually needs, but I don't know a lot about how NAD, for example, would pass through the membrane of the neighboring cell. I don't know if that happens. I don't know enough about that. So I think we'd have to work that out too. But if these molecules that are produced are easily transferable between cells, then you can sort of then kind of walk into a more cell-cell communication. I've got the extra battery, the Energizer battery, and it just plug in, and then that can sort of help the aging cell.
Yeah.
But a mitochondrial genome is a deliverable, like a fresh mitochondrial genome is a deliverable drug.
Mm.
That's probably like a 50-year thing. Maybe this wasn't the right for that. Maybe I should say that one. That's, I don't know.
Or can we engineer a simple bacteria to serve as a mitochondrial? And
Yeah.
mitochondria is an Asian bacteria, right?
Yeah.
Yeah.
So,
Or something in between that gets delivered so that the genome's compartmentalized once it gets into the cell, and then it just kind of boots up
yeah.
as a mitochondria. I like that idea. I'm gonna.
Right, coming out.
I mean, I guess aging people, does that make sense? Would that be enough? Or would that just be kind of cool and still not really? Human?
I guess the assumption there is that the mitochondrial dysfunction at its root is because of damage in the genome.
Mhm.
Might not be true. I don't know if that's true.
And also, many mitochondrial defect, you can fix it through, neuron genes, right? Because they are synthesized by the neuron gene. Doesn't have to be
What's the-
mitochondrial genome just encode like 23 per.
Okay, yeah. So it's I can't remember. I remember this from high school biology. Like, what's the breakdown of genes encoded by the gene versus the mitochondria? And there's quite a few encoded by the gene.
Yeah, most of them are, yeah.
Okay.
So 23 genes, the mitochondrial genome encoded genes are just for some essential function.
I see. So my idea might work.
What other information do you need to build that up?
What's that?
What other information do you need in terms of aging to build that up? Such a large system.
I mean, do you think it would be useful to just be able to target and eliminate synthetic cells? Just very simply, like using a cell-based therapy or using, because if you're talking about gene therapy, if the goal is to eliminate the cells, then you don't care about all of the things that go with, transgenes potentially going into the genome, right? So, I've kind of floated this like a few times since I've been here. I think that, number one is not sure about the chicken and egg question. Senescent cells, are the signals coming from other places? But I think more than anything, it's just that if you're able to precisely and in targeted fashion eliminate senescent cells from either an in vitro model or an animal, then that would be a great thing to study.
Yeah, I think people are working on that, right?
Yeah.
CAR T like way.
Right. The CAR
Yeah, there
T
are
is-
RT based, yeah, I mean, Synolytics also targeting senescent cells, right? But could also have other target if you can identify better Synolytics that kill senescent cells,
Okay.
that would be better. But CAR T cells targeting on some kind of protein on the senescent cell surface. I think there is a paper a couple of years ago.
Yeah.
Well, did it work?
Yeah.
It worked in that paper.
For the limitations.
And it
of
was
that.
public.
Very intense.
Yeah, but it worked in that paper, but I think, so not all senescent cell has that kind of cell
Yeah.
surface, but you don't kill all the senescent cells, right? So you kind of, right now we don't know all the kind of senescent cells. We can kill some, for example, P16 express all, that senescent cell with that specific cell surface protein, right?
Yeah.
But we don't know, I mean, there are other senescent cells which don't have P16, don't have that cell surface protein, right? So, but if synthetic biology can target, for example, multiple markers on the cell surface,
Yeah. Or something more fundamental in the
Right.
gene regulatory program.
Right,
I
right.
think that would be, I don't know, I would as well. But so this was done in an animal, this CAR T, and so what, did the animals live longer? Was that would be?
I think.
Yeah, I
it's
think-
high
yeah, so it
impact
worked.
paper, yeah.
Okay.
It's in Nature or something, yeah.
Oh, it was in, okay, I should have said,
and also, using Synolytics to kill the senescent cell,
yeah,
so they connect something, to the P16, which is senescent cell markers, then fuse to that promoter to kill the P16 expressing senescent cell. So the mouse, doing better, after senescent cell. are
interesting.
removed.
Yeah, we floated this also in the previous breakout room, kind of this targeting the senescent cells and killing them. But we're saying like there are also limitations to that, and like it's probably not enough. Because then if you kill all of those senescent cells, maybe you're like getting rid of a whole organ that's like
Yeah.
Eat up.
so it's like also you need
I
to
used to-
try to like reverse the aging as well to preserve the tissue.
Well, or start it as like a child.
Yeah.
Yeah, so I guess that does kind of point to an interesting dose issue. Do you need to be able to kill all the senescent cells? What is the percent at which it starts to make a
Yeah,
difference?
that's a great question, because senescent cell also necessary and good for some kind of, for example, for wound healing,
Mm.
for some kind of a developmental function. If you kill them, maybe, it's bad, for the future, right?
Yeah.
So what's the dosage, what's the?
It sounds like another controls problem. You need to know when to stop.
Mhm.
Well, it could be there's a meaningful difference between senescent cells that are good, that help with wound healing, and senescent cells that are bad, like that are experiencing replicative senescence or senescence as a result of ionizing radiation or whatnot. So, and we do see that the senescent cells have different markers based on their method of induction. Like if we compare ionizing radiation versus etoposide, even if it's the same cell type, they have different markers of senescence. And so that's a meaningful inquiry if we look and say, could we develop a therapeutic that would selectively target just the senescent cells that we don't like rather than the ones that are helping us? So, like maybe the ones that are age related rather than the ones that are, encoding a tumor or
Is there a marker to be able to differentiate between the two?
my lab has some data that's looking at a lot of different cell types and well, we're just doing etoposide and ionizing radiation to start, but yeah, using mass spectrometry, we can tell like the small differences between them.
Yeah, I guess my question is like, if I were to come and collaborate with you to build a tool that was to say, I want to kill you, but not you. What is that? What is that marker on the, is there a marker on the cell I want to kill that's not on the cell that I want to kill?
Yeah, I mean, well, so I can say that for just the couple of methods that we've tried, and so we don't know for, like, if we looked at, I mean, replicative senescence versus some other type of induction, but based on what I've seen, I'd say there's probably a difference, but of course we'd have to dig in to find out for sure.
I think that's what we would need as some biologists is to be able to.
Yeah, like a verified
Yeah.
difference,
Right.
yeah.
And in the animal model, you can also distinguish between these two types of senescent cell, right? Yeah, that's cool.
So I was writing, so I missed part of what you were saying. It sounded very important. I want to put it in our notes. What is it? You said there's a senescent cell that is good for us and helps us, and a senescent cell that doesn't. So
Yeah.
what? So tell me about the senescent cell that is good for us, because everything I've learned today is that we need to just kill them all.
Yeah, yeah. Some may know more than I do, but yeah, as I understand it, senescent cells also help with wound healing.
Okay.
I think in
Okay.
cases like tumor suppression and things like that. So
Gotcha.
it's not all bad.
Okay. Thank you.
But this is like a double-edged sword, or it is really two different group of cells doing different things.
Right. It's
All the same group of cells doing good thing and bad thing.
right.
That's the
We
question.
we get too many of them as we get older, or if other things too. If we see that in obese patients, they have way more senescent cells than healthy weight patients and things like that.
But the ninth question is very good. We don't know the difference between the good senescent cell and bad senescent cell. I mean, they might be the same, but just function at a different stage, a different need. We don't know the
Okay.
different. Do we?
So that's a good question. How would we, so we know how to make senescent cells using radiation and whatnot. How would we examine senescent cells that are good? How would we induce that from a,
You've only ever been able to induce bad senescent cells,
well, there might be methods for otherwise I don't know about,
but in general, that's probably what it is.
right, yeah.
Well, but I guess have people tried inducing things in a bad way and then seeing if they did wound healing? Like, has that cross model been applied?
Surely somebody has looked at that, but not that I'm aware of.
So basically you radiate cells and then put them in, just see if they are involved.
Mm-hmm.
Know if good senescent cells are damaged, the way that bad senescent cells are induced to be damaged.
I'm not sure. That's a really good question.
Then just think about the damage
Yeah,
piece again.
right.
So-
Sorry, go ahead.
Okay,
please.
so the study, I mean, kind of, in a different direction, they just, kill the senescent cell, for example, during the wound healing and see, it doesn't get healed as well. So they did not, right. They use the PCC markers to kill the senescent cell, but they don't know, the features of the senescent cell. That's my understanding, right?
They didn't induce the senescent cells.
Right,
first.
right. They did
just
not
got-
induce.
Yeah.
I mean, it sounds like at the kind of very basic level, like introducing circuits into, say, an animal or system and getting rid of different types of senescent cells would be a useful perturbation for understanding some of these things.
Right.
Rather than trying to physically remove them,
Yeah.
just administer something that, hits most of the cells in a tissue and then just kills off the ones that are of a certain type. I think that is something, discriminating circuits to discriminate between cell types. I think that's something that
Right.
definitely synthetic biology could think about.
Right, so in fact, I mean, we know there are cells, there are a decision to make, right? So either go into the senescence or go to the cell death, right? For example, this go back to the mitochondria. If mitochondria, kind of open up, there are more DNA coming out.
Yeah.
Then the cell would go to the, senescence. But if mitochondria damage worse, right, cytochrome C coming out,
Oh.
then the cell would go to apoptosis.
Yeah.
But But if it goes to apoptosis, that's good, right? The cell's gone.
I see.
But go into senescence in the future. I mean, if you can do synthetic biology to control that step, right?
Yeah.
Just, make the mitochondria release the toxin to kill the cell.
Yeah.
Then you don't have the senescent cell. Maybe that's good for aging, right?
No,
That's
good.
maybe feasible for synthetic biology kind of, approach.
I mean, the immune system should be running around pruning these senescent cells, right? And breakdown in that function is part of why they accumulate.
Apparently, I mean,
No, okay.
the other way, the senescent,
Wikipedia is wrong.
yeah, the senescent cell actually release some,
Okay.
cytokines that will cause the
Okay,
misfunction of the immune system.
interesting.
And so
Okay.
I just want to follow up on something, so, we basically have this sort of cell that's either gonna undergo senescence or apoptosis. And that decision is basically dependent on the health of the mitochondria.
For that specific model, it is,
Right, okay. and
So the cell have to make a decision.
Sure.
yeah.
And so you then said this is a really good place for synthetic biologists to come in, and so is your idea that there would be some way to, like an AND gate or gate, some sort of gate
Oh.
where it was like, okay, if the cell is producing this molecule, then we make the decision to die, basically,
Right.
rather than senesce. Is
Right,
that
so-
what you're thinking?
I think because, the whole thing, whether the mitochondria release thing more damaging or not, has something to do called, mini amount, based on its permeability of the mitochondria, right? Just more permeable, things,
Okay.
to come up. So if it's a mount, is a very permeable, the cell, the things, getting out of the mitochondria based on cytochrome C and other things that would induce apoptosis signal, right? But if it not, that normal things, normal, permeability, right? So, but if it's they call the mini, minority, amount, that means, something, DNA would come out. DNA, maybe other things come out that would induce senescence, right?
I see.
But that permeability maybe, can be controlled
Sure.
by synthetic biology, right? So based on just control whatever function
Yeah.
in the, mitochondrial membrane. Right? So if that part can be controlled, maybe, either, mitochondria can cause, more damaging, make cell die, or more normal, right?
So you're saying preventing senescence in the first place by taking every time a cell makes that choice, pushing it towards apoptosis instead of senescence?
Right.
Would there be a problem with that if you did that across the whole body?
So, he mentions, right, senescence, senescence cells is tumor suppressing.
I know. That's the thing, do they all come through that way?
Right, that could be
Yeah.
bad, right? So it need to be studied, right? Senescence, if it function as a tumor suppressor in that specific tissue or at that specific stage, if you don't make cell